Today, I would like to talk about in silico drug discovery for COVID-19 using gene expression profiles of mouse infected by MHV and those of human cell lines infected by SARS-CoV-2. My name is Taguchi from Department of Physics, Chuo University, Tokyo, Japan. This is collaborating work with Professor Taki from King Abdul Aziz University, Saudi Arabia. This talk is in 7th Annual Congress of the European Society for Translational Medicine of COVID-19. Today, I would like to talk about the contents of two recent published preprints. Since I don't have enough time to explain the contents fully, the people who are interested in the details of this study, please refer following these two preprints. These are two data sets I analyzed in this talk. First one is in vivo dataset of mouse infected by MHV. MHV is analogous coronavirus, which is infectious to mouse. There's a two datasets, 3 days after MHV infection and 5 days after MHV infection and control. There are tissue, liver and spleen, and there are also two distinct genotypes. The purpose of this study to identify genes expressed distinctly between control and infected mouse, independent of the tissue and genotype. In addition to dataset I also analyzed the in vitro dataset of human cell lines. There are 5 human cell lines infected by SARS-CoV-2 or corresponding control human cell lines. The purpose of this part is identify genes which is expressed distinctly between SARS-CoV-2 infected cell lines and control independent of 5 cell lines. Gene expression of these datasets are formatted as tensor. Since I don't have time to explain what the tensor is, if the people are interested in what the tensor is, please refer to my preprints. XIJKMNP represents the expression of ICE genes either of 2 genotypes and either of 2 tissues, and if it is control sample or infected mouse. And for these conditions, there are 3 biological replicates times 2 technical replicates are available. For dataset 2, tensor is much simpler. It has only 4 suffixes. XIJKM represents the expression of ICE gene of 1 of 5 cell lines, and it is infected cell line or control. And for these conditions, there are 3 biological replicates. With some mathematical trick, we can decompose these tensor into the product of 6 vectors or 4 vectors. Each of 6 vectors represents the dependence upon genotype or tissue, or if it is control or MHV infection, or biological replicates or technical replicates or genes. These vectors are not unique, but represent several dependencies upon genotype and tissue and control and MHV infection, and biological replicate and technical replicate and genes. Then the several combinations of product of these six vectors is summing up to x, I, j, k, m, and p with the weight of g. Similar procedure can be applied to the dataset 2, and the four vectors attributed to the dependence of cell lines and SARS-CoV-2 or control or biological replicates or genes. And there are also multiple dependence upon cell lines and the SARS-CoV-2 control or biological replicate genes. Several combinations product of these four vectors are summing up to the xijkm with the weight of g. First, we need to find which vectors attributed to genotype and tissue and biological replicate to technical replicates independent of genotype or tissue, biological replicate, technical replicates, because we would have to find genes expressed distinctly between control and MHV infected mouse, independent of the genotype and tissue and biological replicate or technical replicates. Then we found first vectors attributed to the genotype and tissue and biological replicate and technical replicates are independent of the genotype and tissue and biological replicate and technical replicate. On the other hand, second vector attributed to the control and MHV infected mouse represent the distinction between the control and MHV infected mouse. So we decided to employ the combination of the first genotype vector, the first tissue vector, the second control and MHV infected mouse vector, and the first biological replicate vector, the first technical replicate vectors. We need to do similar things to dataset 2, and we have found first vector attribute to the cell lines are independent of the cell lines, and the first vector attribute to the biological replicate are independent of the biological replicates. And the second vector attribute to the SARS-CoV-2 infected cell line and the control cell line represent the distinction between the SARS-CoV-2 infection and the controls. So we decided the combination of the first vector attribute to the cell line and the second vector attribute to the distinction between SARS-CoV-2 infection and the control, and the first vector attribute to the biological replicate. Next step, we need to find which vectors attribute to the genes associated with a combination of the vectors identified in previous slide. For dataset 1, we have selected the first, second, first vectors, and we have found third vectors attributed to the genes have largest weight. So then we have decided to employ third vector attributed to the genes for selecting genes. For dataset 2, we have found fifth vector attributed genes associated with the combination of the first, second, first vectors. Then we have decided the fifth vector attributed gene to select genes. P-value is attributed to the genes with assuming these vector third vector attributed genes in dataset 1, the fifth vector attributed genes in dataset 2 obeys Gaussian, and using cumulative chi-square distribution, we attribute the p-values to gene I. Then these p-values are corrected by Benjamin-Hohenberg criterion with assuming multiple comparison correction. Then we have found 134 genes are associated with the corrected p-values less than 0.01 for dataset 1, and 163 genes are associated with the corrected p-values less than 0.01 for dataset 2. Then we have uploaded the two sets of genes to Enricher. Enricher is a famous enrichment server which can validate the biological meaning of uploaded genes. There's a two biological validation of uploaded dataset using enricher. First one is if the uploaded genes significantly interact with the SARS-CoV proteins. Second validation is if genes are altered by a SARS-CoV infection. After these biological validations, we also try to find drugs that affects the expression of genes we have selected, because such drug can be a potential candidate drugs for COVID-19 treatment. This is the result of the first biological validations of uploaded genes. So for dataset 1, we have found 4 SARS-CoV proteins are significantly interacting with the genes we have selected. For example, the 194 genes and human genes are known to interact with this SARS-CoV protein, and among 194 genes, 10 are included in our selected genes. This is just the p-values. It is obvious these 4 SARS-CoV protein are interacting genes we have selected significantly. For dataset 2, we have more SARS-CoV proteins which significantly interacting genes we have selected. So these are corrected p-values, and it is obvious these SARS-CoV proteins are significantly interacting with genes we have selected. So from this point of view, our selected genes is biologically reasonable. Now this is the second validation of our selected genes. For dataset 1, 6 SARS-CoV virus significantly affects the expression of genes we have selected. For example, 300 genes, human genes, are known to be affected with the infection of this SARS-CoV virus. And among 300 genes, 10 are included in genes selected by ourselves. Now, this is the corrected p-values, and so it is obvious these 6 SARS virus affecting the genes we have selected. For dataset 2, we have more SARS-CoV virus which can affect the genes we have selected. These SARS-CoV virus upregulated genes we have selected when they infected the cell lines. And this is a SARS-CoV virus which upregulated the expressions genes we have selected. These are corrected p-values, and it is obvious these SARS-CoV virus affect the expression of genes we have selected. So again, from this point of view too, our selected genes are reasonable. Since we have confirmed the suitability of a selection of genes, we have tried to find drugs which is effective to SARS-CoV-2 infection. First, we have compared our result with drug matrix. Drug matrix stores the gene expression of rat tissue treated several compounds. And we found these 3 antivirus compounds. So unfortunately, we don't have enough time to explain what these antivirus compounds are, but these 3 compounds significantly affect the expression of gene we have selected. For dataset 2, we have also found 3 antivirus compounds affecting the expression of genes we have selected in drug matrix. So our strategy successfully identifies drugs which can affect the expression genes we have selected. We have repeated similar procedure to the another data set in which the drug perturbation from gene expression reverse, and we have selected these antivirus drug that affect the expression of genes we have selected. And we also try to find gene drugs that affects genes we have selected in the links else worse than chemical perturbation, and we have found several antivirus compounds affect expression of our genes. So our strategy clearly successfully identified drugs which can affect the expression of selected genes, and these drugs might be effective COVID-19 treatment drug. So although we don't have enough time to explain compounds identified one by one, I would like to introduce one example. The example is ivermectin. Ivermectin is once found by the famous Japanese Nobel Prize winner scientist Professor Omura as a parasite-killing drug. Now in Japan, ivermectin is under the clinical trial to check if ivermectin can affect the SARS-CoV-2 affected model animals. So this means our strategy can successfully identify drugs which can be useful for COVID-19 treatment. So that's all I would like to introduce here. But before closing my talk, I would like to also compare my result with two gold standard datasets. First gold standard dataset is SARS-CoV-2 protein interacting proteins. So here the SARS-CoV-2 proteins, and this p-value means these SARS-CoV-2 proteins significantly interact with the genes we have selected in dataset 1. And this is odds ratio. So you can see odds ratio is very large. And for dataset 2, we have also found all SARS-CoV-2 proteins are significantly interacting with the genes we have selected. So again, the odds ratio is very high. So at least for comparison with gold standard SARS-CoV-2 protein-protein interaction dataset, our genes is useful. And another gold standard dataset is a dataset of possible drug repositioning list for SARS-CoV-2. And here's drugs included in this list. And the number is the number of experiments where these drugs significantly affects the genes we were selected. So now you can see there are many experiments where these drugs can affect the genes we have selected. For dataset 2, we have also found many experiments the drugs in the gold standard dataset can affect the genes we have selected. So also from this point of view, our selection is very reasonable. The conclusions. We have identified two sets of genes whose expression altered by SARS-CoV-2 in vivo, in vitro, respectively. Two gene sets are coincident with non-SARS-CoV protein interacting human proteins and genes whose expression altered by SARS-CoV infections. And drug repositioning, including ivermectin, performed with identifying drugs that affect the two sets of genes. Then finally, we have done two comparison with gold standard dataset. One is coincident with SARS-CoV-2 PPI, and second, the drug repositioning proposed for SARS-CoV-2. So based upon that here, we can conclude our strategy might be promising procedure to identify drugs which are effective for COVID-19 treatment. Thank you very much for your interest.